Hello students, in this video we are going to discuss applications of biotechnology in the field of medicine. In the earlier video we discussed about applications in the field of agriculture. Here we'll focus only on medicine part. Now the first application could be making use of genetically engineered insulin. Now this word insulin we usually hear when we look at any patient who is suffering from diabetes. What happens with these people is that the insulin level in their body becomes very low, or their body is actually resisting insulin that is getting produced in their body. Now what is insulin exactly? See, whenever you are eating something, for example carbohydrates, those carbohydrates get converted into glucose, and this glucose is nothing but a kind of sugar. So whenever you're eating something, the sugar level in your blood will get increased. Now, if it is increasing, it is not a good thing for you. So we have a special organ in our body that is called pancreas. This pancreas produces a special hormone called insulin. And this insulin controls the blood sugar level. So I'm saying if you have insulin in your body, it can control the sugar level in your blood. So it will not increase beyond a certain limit. It's a good thing. Now, in case this insulin level has dropped in your body. So there are patients who complain that our insulin level has dropped. It has become very low. In that case, doctor treats them for their disease called diabetes. Now, what doctors usually do? They take the insulin from a different animal. They take from pigs or cows. So just like we humans have pancreas, even pigs and cows will also have pancreas. And they also generate some kind of insulin as a hormone. Now the point is, it is not the same insulin that we generate in human body. It can be little bit different. But doctors make use of that particular insulin and put it in the body of humans. Now, some patients, they say that they are feeling better, they recover from this problem of diabetes. But there are some patients who say that we are facing some allergic reactions. The insulin that you are using is not suitable to our body or not suitable to our blood. Now, in that case, we say that we cannot use this insulin which is coming from other animals. We need insulin that is getting produced in a human body. Now that's a big problem for us. And second problem is the process is very time consuming. If you're bringing some insulin from a different animal, the process is very time consuming and it is very expensive also. So now making use of the biotechnology, making use of this genetic engineering, we are generating insulin which is sourced from human body. So what we are doing? We will take a sample from our human body. So we will take a sample of insulin. So we will take a special gene out of this insulin, a gene which is making this particular insulin. And we will put that gene in a bacteria. Now the name of this bacteria is E. coli. We can make use of other bacteria also, but usually make use of E. coli. We will put this gene inside this bacteria. Now here we are doing genetic engineering. Now what will happen? If there is a gene which can produce human insulin, then if we have added it in the bacteria, even that bacteria will start producing the insulin which is of human origin. So now what we will do? We will increase the number of bacteria. We will grow this particular insulin inside the bacteria. And later on, the same insulin will be brought in our body or the patient's body. So now the patient will get insulin which is not originated in the pig or cow, rather it has origin in human, so it will be suitable to human body. It will not go ahead with any kind of allergic reaction as such. So here we have made use of genetic engineering. And the process that we follow over here, or the technology that we are following, is called as recombinant DNA technology. Recombinant. Why we are calling it recombinant? So here we are actually combining two different DNAs. So there was one DNA which we took from a human body, and there was another DNA of this particular bacteria. So what we did, from the DNA of the human being, we took one gene and introduced in the body of bacteria. So the point is, because there are two different DNAs involved over here, that's why we call it recombinant DNA technology. So it's a good technology and we are generating genetically engineered insulin. A good application of biotechnology. Okay. Now we have second application in terms of gene therapy. You must have observed there are some diseases which are called as genetic diseases. So the reason behind that disease is your genes. So for example, sometimes people complain that I have got asthma, and doctor says that you have got asthma because it is part of your gene. Someone among your family members must already have this kind of disease and it has got transferred to you in the form of genes. So the origin is genes. Now, if a person has got any such disease, that is most of the time that is due to some kind of defect in the genes. What happens is that genes are getting copied, and when they are getting copied, sometimes a defected copy is generated, and because of those defected copies, many times some diseases are taken ahead in the body of the other generation. So the point is, because there is some problem with one particular gene, that's why you have got one disease, and sometimes we call it as a genetic disease. Now what we traditionally used to do, whenever you used to go to doctor, they used to do one thing: whatever are the symptoms, they are going to address those symptoms. Let's say you have said my hand is getting red, my eyes are getting red, so they will just address those symptoms. But please understand, the core issue is not getting resolved. The core issue is there is some problem in our genes, that there is one gene which is defected, which is mutated, so it is not the correct form of gene. That is the core issue. So the issue is not resolved completely. Now we have a solution from biotechnology, wherein we go ahead with gene therapy. What we can do? Making use of genetic engineering, we can introduce the correct gene in the body of the patient. So you said that you had one gene which was defective. So we can do one thing: the copy of this defective gene which is not defective or which is in the correct form, that can be introduced in your body. So we will take one cell, we will add it in that cell and then reintroduce that in your body. So I'll give you one example. So let's say there is a patient who is having deficiency of ADA. ADA means adenosine deaminase. Now this is a protein. Sometimes what happens is that some people are not able to produce this particular protein in their body. So this is a defect which is genetic in origin. So there is a gene which has to produce this particular ADA and it is not producing. What we will do? We will take this human being's blood sample. Now from this blood sample we will take a cell and we will make a change in that particular cell. We will add or insert the correct form of this particular ADA, adenosine deaminase, and then we will put this particular blood sample back to the human body. So now the issue is completely resolved. It was genetic in origin and you have made changes in the genes itself. So now we can say through this gene therapy, the issue gets resolved completely from its base, from its core, right? So this was the second application. Now the third application is molecular diagnosis. Now sometimes some patients, they come up with their problem, but the problem is that they have detected their disease after a very long time period. What happens is that by the time you see the symptoms of the disease has already become very big, very large, very widespread. So we are saying you have detected the disease very late. You have been very late. If you could detect the disease in early stage, we could have resolved it very easily. But now because you have come up very late, the issue has become very complex. We are not able to save you from this disease completely. This is a problem. Now traditional method used by doctors for the diagnosis means they are checking what is the exact problem. They will ask you 100 100 questions. They will check your x-ray, they'll take your MRI, they'll check with the microscopy, they'll go with your blood sample, urine sample, etc. All these things are done only for the sake of diagnosis, to come to a conclusion what is the core problem in the body of that patient. Now we have another solution in the form of molecular diagnosis. What we can do is that instead of getting late, we can diagnose the disease a very early. Diagnose means we can detect the disease. How we can do that? You must have heard about this PCR, polymerase chain reaction, or ELISA, enzyme linked immunosorbent assay. These you must have heard at the time of COVID-19 pandemic. We were doing these tests, ELISA test or RT-PCR. Now, these tests are nothing but making use of molecular diagnosis, wherein you can check in a very early stage whether you have got this disease or not. How they check? They go to the molecular level. Now, what they will be doing exactly? For example, if you take the example of this PCR, what PCR does? Let's say there is a virus which has infected your body, but the virus is in very low quantity, so you are not able to detect the disease. So when we go ahead with this PCR test, what the PCR will do, it will generate multiple copies of your DNA or RNA, multiple copies. So the virus which was in lesser quantity, now you're getting multiple copies of it, so its quantity will be increased. And once the quantity is increased, now you can detect whether you have got the disease or not. So the point is, the detection becomes easier. Or let's say if you're taking the example of this ELISA test, what this ELISA test does, it just checks in your body if there is any antigen or antibody exists or not. For example, let's say the COVID-19 virus has got infected to me. Now after infection, I did not feel anything and my body fought against it. So COVID-19, the virus has got into my body. After that, my body generated some antibodies to fight with this virus, and the issue got resolved completely. I did not even know. But after some time, I did this ELISA test. This ELISA test checked whether my body contains some antibodies which can fight COVID-19 vaccine, and it found it. So that means I had this disease called COVID-19. COVID-19, I was infected with it. So the point is, we can know whether we have got this particular disease or not in the very early stage, making use of this molecular diagnosis. At the molecular level, we are diagnosing it. At the very smallest level, we are checking it. Clear? So these are the three methods or three applications of biotechnology in the field of medicine.